never ending.
Exactly.
I mean, the summers are great and you have a very long summer, but then pretty much end of October, November until March, it just rains non-stop.
I don't even care for the cold or the wind really, but the rain, it's just so annoying when it doesn't stop. It just doesn't stop, especially with the storm now. Like it's storming in France a lot, so.
And London, I could always kind of sneak through the rain.
Yeah.
Then I moved to Bordeaux and I bought the full outfit because there's no escaping.
Chris, where are you? Why are you leaving me alone here? Hello?
He's home, scared of the rain.
Yep.
Yep. Not even denying it.
But you're still coming, aren't you? Later?
Maybe.
Oh.
So no.
I'm going, Anna.
Cool. At least someone.
Yeah, I'm already in Bordeaux, but it was raining, so it's better to.
It won't stop, Marcella. It won't stop. That's not an excuse anymore.
I know. But the plan was to attend Neuroccino while walking, but not possible with the rain. So I said, okay, I will stay here. I will attend Neuroccino at home, and then I'm going. It's fine.
At some point, you guys got to get over the rain.
I know. I'm prepared. No worries.
Okay. Right. So a couple of new people in the room. Let's just very briefly spend some time on the ground rules. So everyone is welcome to join us and present a paper, a book, or a piece of science they thought was interesting. And we now are very organized. We have a rota. So there's now, thanks to Anna's initiative, there's now a schedule of who's presenting when. Did we include Hervé in that schedule?
We didn't, but I mean, no, actually we did.
So if you could just share that in the chat so everyone can see the rota and when it's their turn. I think I'm next on the list. So there's a breather for everyone else. But we have a rota, and if you need to swap your week, we can always do that. Don't try to force it if you really don't have the time.
Okay, of course included. Of course, he's there.
Great. Great. So today we have Sabrina, who prepared 2 papers. Sorry, one. I sent her one that I got really excited about, but she already prepared one, I think. So I don't know which one it's going to be today, but if you share the link in the chat, the floor is all yours.
Okay, thank you, Stephanie. Good morning, everyone. Well, yeah, so at the end, I just present one. So here in the chat, there is the link. Okay, and now I go to share the screen. Well, better in this view. And just fix. All right. Okay, so as you can already understand from the title, today I'm going to present this article on this new cellular atlas, which brings us really interesting and useful data. So they obtain a cell census of the broadcast area of the human brain at micrometer resolution. And let me check the day. Yes, it was really recently published. We can see here, so in October. And as we can see, a large group of specialists collaborated, and we can read all the names. So it means that there is a great collaboration of different institutes. We have Massachusetts General Hospital, Boston University, the European Laboratory of Nonlinear Spectroscopy in Florence, and the Icahn School of Medicine at Mount Sinai. Okay, so I'm going to give you an overview based on what is written. And given the complexity of the data, I will not go into the methodological details. So I invite you to read the article for more data and details. Well, in the first part, I'm going to the introduction part of the article with some information on what exists until now, and also the limit of the technology that is with this paper is overcome. And then in the second part, I report the approach used with the reference to figures and a video. And then in the last part, in the third part, I give you some considerations written in the paper. Okay, so let's start. Important steps have been taken so far regarding the human brain. So we have several 3D models with the cellular resolution that have been created using different methods. For example, here they report the Big Brain project that with 5 years of work, they obtain a comprehensive data set of a human brain with high resolution. We also have different brain atlases and different waves of data analysis of large data set, but the technology until now have a limit. So they cannot visualize the microscopic features of the brain without a distortion. So it does not allow accurate visualization of the morphomolecular properties of the tissue. So for example, individual cell across different regions or laminar cytoarchitectural boundaries. So they overcome the limits of each individual methodology using different innovative images and analysis techniques. But how? With a multimodal approach, so overcoming the resolution gap between macroscopic and microscopic techniques, and integrating the cellular anatomical information within our brain reference space. And where they considered the Broca area with the reference to Brodmann area 44 and 45. So with their approach, they managed to collect the cellular census of different neural types in Broca area with the MRI reference system of the entire hemisphere. They worked on the postmortem brain of a man with 79 years old who died of natural causes, so without clinical diagnosis or neuropathologies. But let's go on with the first figure. Here we have a clear summary of the complex work. So they use different imaging techniques, MRI, OCT, optical coherence tomography, LSFM, light sheet fluorescence microscopy, and the 3D stereology on LSFM. Okay, so they registered and analyzed the data of the different modalities with each other to obtain a special reference at the macroscopic level of the laminar and cytoarchitectural boundaries, and to obtain a cell count at a microscopic level. So with the aim to have the cellular census of Broca area at high resolution with the spatial reference of the entire brain. I have a video to show you. So that is a video that you can also find the supplementary material of the paper, which I found really useful to understand the different modalities. So in the meantime, I'm going to share. Well, we are going to navigate across the sagittal MRI slices. We'll see the coregister slices of OCT LSFM, and also we'll see the manually segmented cortical layers. So you should see a black view. And now let's go on. Okay, let's come back to the paper now. Okay. So now we are going to see its technique step by step with a few figures. Let's start with the MRI. So with the MRI, we have a macroscopic MRI reconstruction of the whole brain. And here we have an example of the distortion correction. We can see the vessels here on the left, but on the right with the correction of the distortion. Well, it's quite clear. And here, well, I go directly to this nice image of an actual view of the brain, and then of the sagittal view. I hope also that with Zoom you can see well. Otherwise, you can see directly the paper. Okay. So what is the second technique? OCT. Well, this is an intermediate modality to enable the registration of the distorted histological volumes containing the cell type information to the MRI coordinate system. So it's between this one and this other modality. But let's go on to see a few images. So here we can see a slice of the OCT volume intensity and a zoom to see the height quality. And and here the blood vessel segmentation of the OCT volume. Then the third one is the LSFM. So with this one, you have a microscopic volumetric histological imaging of single slabs. And I didn't say that with MRI, you have a whole hemisphere. And then with the OCT, we have the slices. And then with the LSFM, we have the tissue slabs. And then another thing is that they did a nonlinear registration of these three modalities from this one, so LSFM to OCT, and then from OCT to MRI. So this is the registration. Now let's go on to the last technique. So where they obtain a cell type specific quantitative census of the neurons. So in other words, it's a work of cell counts and is a manual labeling of the features. But before, we didn't check this one. So the LSFM. So let's go directly before to this image where we can see the vessels in blue and in green. And then on the right, you can have the different cellular markers. Well, I zoom so you can see. And this is. And then, yes, as I said before, let's go to see the figures of the last technique. So here, well, the first one is also interesting because we are going to see a slab, but the systematic random counting grid using during the stereological analysis. Interesting. And here we can see the different data levels. And well, in the different data levels, we see the neurons. And here on the right, we have the estimation of the total number of neurons. Here we have one type of neurons, this one. And then here another type of neurons that, as they write here, they only consider the layers 2 and 3. So for this, we see just here the neurons. Okay, with the last figure, we can see the sagittal view of the brain, left hemisphere, and then a zoom to this area. In particular, they selected the area between the pia surface and the white matter surface. And here, with these lines, we can see, there is okay, now it's better. And here with these lines, we can see 10 surfaces and the markers in green. Okay, so now I move on to some final consideration that they reported. Oh, but before a first useful information, because you can have the data and the atlas in the Dandy platform. You can get the link from the paper when you open the paper online. So it's easy to have access, I saw. Okay. Then, yes, here, this data and this single cell and spatial cell approach are helpful in the study of brain disorders. So helping to understand the pathogenic and the compensatory mechanism. And here the name few disease, as we can see. Just to say how it could be interesting also to understand better also these different situations. And then last but not least, they say on this other side that if we work on axonal markers and myelin markers at this level of resolution, it will help also the DVI studies. So therefore the knowledge about connectivity. That we know in this field is useful to know the direct measurement of axonal orientations. In particular for the area with a lot of fibers. Okay, so yeah, from my side, I find it's really interesting to be able to see a brain area with such a high resolution and the possibility to reflect on the use and application of these data and methods. Of course, all this takes time. In fact, it was a long work on just one part of the brain, the Broca's here in this case. But, yeah, so it's something to consider in the organization of new works. But step by step, perhaps we could arrive to apply this work on the whole brain. But yeah, so anyway, in the meantime, they don't waste time because they are already working on the same method on multiple subjects. So these that I said before. But I add that by reading an article on the CNR website, so the Italian National Research Council, it was a press release. I found that this international group is also going to work on the brainstem. So they received funding from the National Institute of Health, and the aim, well, I have a note, so I can say specific. What is the aim? So from Italian to English. So they are going to resolve, they say, at a microscopic level, not only the types of cells present, but also the pathways and the neural networks that characterize it, and the connection that establish the brain-body functioning. Good luck with this work also. Okay, so here I conclude my presentation. And I'm also interested to know what do you think of this work, what they did, and the future directions that we can have. Thanks.
Thank you, Sabrina. Yep, questions are in. Chris, first one up.
Yeah, thank you for the presentation. I was wondering, because there are different definition of what and where the Broca's area is, right? So how did they just choose a definition of this of the area? And did they make sure that it makes sense at this level of resolution? Because if you define it with macroscopic landscape features, that might not be relevant at the microscopic level, and or if they did the opposite, is it relevant at the macroscopic level? And then is it easily identifiable between subjects? So I know that here they only used one subject, but if they want to expand, as you showed, they want to do it on 10 subjects. How is it comparable? How easy it is for them to basically register all of them on the same area with their definition of the Broca's area?
Yeah, good point. I also saw about this, but what I found is here that they say that the Broca's area is the based on the Brodmann's area, the 44 and 45. So I think they went directly to this area based on just the Brodmann's classification. So just reading in general the paper and the first part, I found these informations. I don't know if maybe there is other things, because also I didn't say that there is, after what we saw, another part here, the material methods. So with more and more information of each techniques, and maybe also how they decide and delineate also the area. But otherwise, until now, I didn't see. Well, here you have also the supplementary materials referment. But yeah, it's absolutely interesting also this, but yeah, seems based on this.
And how do you think it would generalize to multiple subjects? Also, the Brodmann areas or the Brodmann segmentation is quite old, so I don't know if it stands to the evolution of neuroimaging in the last decades.
Mm-hmm. But I think the idea was to start with area and to see if the method works. And yeah, interesting that they decided to the Broca's area. And then thinking to this other work with 10 subjects. Well, I think they're going for each subject to find, based on the anatomy of the subject, this area, based on the information that they have based on the identification of the 44 and 45 areas. So in this way, they can have the same area based on the anatomy of each subject, and then compare, in particular, I think, of based on its anatomy, but then the number of neurons, because then, as we saw, they arrive, or we have this resolution of the brain, entire brain, but then the count of the cell types. So maybe they can go to see a different number, I don't know, and we can have a view and see the variability based on the different subjects, and based on with the number, total number, and also the types. Maybe we could see if there is any different also in the cells that we can have in this area. Then also interesting that they are going also in the meantime to check also this method with another area, the brainstem. So in this way we can also have a comparison of different area, but we can see also the same method with another area. Yeah, and let's see.
Right. Thank you. Eduardo has a question for you as well.
Yeah, thank you. Well, they relate more or less to what Chris already asked it. My question was, why did they choose Broca's areas? Is there any specific reasons? And why did they choose to focus only on the left hemisphere? I know it's time consuming, but whatever. And also, why did they choose Brodmann definition of areas? Because as was already mentioned, Brodmann atlas is kind of old, but that's not a major problem. The problem is that we don't even have the original data, so it's kind of tricky atlas to use.
Yeah, good other point. Well, if I'm not wrong, Brodmann, well, let's go here, yeah. Brodmann's atlas is based on cytoarchitectural informations. So I think this was a connection to, so it's this one. And then I don't know why they choose the Broca's area. And then, yeah, another point is that, well, if you say Broca's area, we know everyone what is the area, but then there is a lot of discussion, where is this area, in fact, as we said. So, so you can have a bit of confusion also to say every time just Broca's area. But since just reading the part that I read, that's, yeah, for the in fact is just this area.
If I can jump in here, I think they actually solved it quite nicely, because they specify that in this work, what they consider as Broca's area is BA 4445, and most papers don't specify that. They just talk about Broca's without actually saying how they consider Broca in that case. I think in this particular instance, it's justified because literally the first sentence is, in this work, this is what we mean. So if you if you define it, it's fine. And then there's probably various parts of the brain that you could have chosen to do that study, but then the inferior frontal gyrus on the left side is a hotspot that everyone is interested in, and it's cytoarchitecturally well defined. It's anatomically well defined. It's somewhat functionally well defined, and it's an extremely important hub for speech and language. So I guess having further detail on that particular part of the brain is definitely of a wide interest. Now, that said, starting with a complex area like that is always a bit tricky. So for me, it would have been interesting to also see the difference to primary cortical areas like M1 or S1 or even V1 in comparison, just to get a better feeling. But that's just me jumping in. Eduardo, do you still have a question? Did that answer your question?
Yeah, kind of. Yeah, I was just curious about these kind of details. And also, what do you think about how useful this can be, like to have such a precise spatial description of an area in the brain, how it can be useful?
Well, as we know, also we have also few disease where it's based on one type or the other type of neurons, and also sometimes also specific to just one area. So maybe we can have other information to better understand the clinic field, having exactly more high quality in the more informations on this, so on the cells, and then with the referment to the special with the coordinates of the brains. Yeah. So in this way, you can also go to check every time also in the others situation, how is the situation, and have as a base these informations. So could help.
There's a big push for higher resolution data, and we're in one of the hubs with the 14T scanner that's coming our way. So there's definitely a need for a better anatomy of the brain, but I was also wondering if they offer the maps they created in the study in a platform like eBrains, for example, so you have multiple layers of information in the same reference framework. Because then you could really combine the cytoarchitecture and the cellular information, functional information, all in one place in theory.
Yeah, right. I also had that they used, I read, 7 Tesla, same as MRI scan in this work.
Which by current standards is high resolution, high field imaging. So that's perfectly legit. Annie has a question as well.
Hi, I was basically already brought up by most of what you now said before me, just for the bigger picture, that for now, because it ended in what this might be useful for, right? And we're wondering the same at the moment, because they didn't mention how this is related to any function, but I guess that would be for us. I mean, of course, it's always interesting and a push for micro, but for me, there's always, if there's no relation to any function, then yeah. I would love to hear, similarly to what you just mentioned, if there's any idea to relate this to any functional measures they might have. Of course, as I understood, and that's my main question, this is all only applicable postmortem. Those really micro, the methods you mentioned that most of us hadn't heard of. So for now, all we can relate to is the pre-mortem function of the human.
Mm-mm. Well, yeah. Well, for now it's just post-mortem, so, and we can't apply to the brain in vivo, but yeah, so until now we can arrive to have just an atlas, or information, so yeah, just a way to understand better the brain, but yeah, of course, there are other aspects then to consider.
Before I let you go, there's actually 2 questions on YouTube that I'm just going to read to you. Do you think that cellular resolution imaging could be helpful for providing information precisely against the structural lessons or tumors? Can it assist or assess the amount of damage done? And then I don't know what that word is supposed to mean.
Well, as we saw, also we can see better the vessels.
It's actually specified after. Can it lead to preserving the function of that particular area if we have had cellular imaging guiding the precise nature of a tumor or a lesion? So I guess what's the added value of cellular imaging in terms of those? It links back to what Annie basically just said. If that's really an added value given what we already know from lesion and tumor studies.
Yeah, well, I don't know in specific about cells. I not also work with the cells. But I can say that as we saw also, also high resolution of the vessels with these techniques, and we can see and go maybe to understand better also the vascular or have more information also on this. And we know it's also important for this disease. And then also I like the connection to DY studies, because they say that if we apply these methods to the axon and, well, the white matter in specific, maybe we can arrive to understand better also the directions. And we know also for tumors, there is a big involvement also in particular of the white matter, also in stroke, but we know it's really consistent in for tumors also. So we can better understand the situation of the brain, how is, for instance, also in the posterior temporal area. We know we have, many tracts, fibers, so it's quite difficult sometimes, or also in general, to select and the specific tract. So maybe this can help, these methods.
I guess it's always important to bear in mind that anatomically speaking, we want to go and map every single inch of the brain and know exactly which cell is where and what does what. And pushing the resolution and pushing our methods goes in that direction where you really buckle down and you get to smaller and smaller units to understand the structure of the brain. But then when it comes to the function, we know that the function doesn't restrain itself to Brodmann areas or gyri necessarily. So I guess it's always this trade-off between acceleration and going deeper and deeper and looking at the tiny structures in the brain, but also taking a break every now and then and look at the bigger picture and the surroundings to map the function along with it. And then also maybe some of the functions that we have are just too complex to pin them down on individual structures, which we know is the mapping problem. So you have individual linguistic functions that you can't just put on one cell type, very likely. We will see in the future, but for now, it's unlikely that you have a one-to-one match here. Right. I don't see any more questions and we're way over time. So thank you very much for presenting that paper, Sabrina. I wish you all an exciting week in science, and next week is my turn and I don't know which paper yet. So that's going to be a surprise. Have an exciting week. See you next week. Bye-bye.